Hi, I'm Sean Mooney, professor at the University of Washington and Chief Research Information Officer of UW-Medicine. Over the past 2 years, we have been creating an initiative in medical data science, and as part of that, we have created this seminar. Speakers will include world-leading scientists who will tell us about how data can impact human health and their healthcare. Please use the link below to register for our mailing list to hear about future events in this series. We hope you enjoy them.
Great. So it's my great pleasure to introduce our two speakers today. This is the first of the seminar series for this academic year, as we don't tend to have speakers over the summer. And as the chair of the Department of Biomedical Informatics and Medical Education, which is the administrative home for this multidisciplinary center for medical data science, which has faculty leadership from engineering, public health, and medicine. It's also my pleasure to kick this off because the topic today, which is computational variant and genome interpretation, a model for evaluation and calibration of machine learning for clinical decision-making, is near and dear to my heart as someone who's been engaged in the field that's now known as translational bioinformatics since its inception. So this first speaker will be Vikas Pejaver, who is an alumnus of our program. So I've had the pleasure of working with him in the past. So I guess for this time, Sachi, he'll have to count as our quote internal unquote speaker. Vikas is currently an assistant professor at the Institute for Genomic Health and the Department of Genetics and Genome Sciences at the Icahn School of Medicine at Mount Sinai, with a research focus on developing and applying machine learning methods to link genetic variations, molecular function, and disease phenotype. He received his master's and doctoral degree in informatics from the School of Informatics, Computing, and Engineering at Indiana University in Bloomington, came to the Department of Biomedical Informatics and Medical Education at the University of Washington, working with Sean Mooney, the head of the Institute for Medical Data Science, as well as working with the eScience Institute at the University of Washington. And during his time here, he received a Moore Sloan and Washington Research Foundation Innovation in Data Science postdoc fellowship, as well as receiving a K99R00 grant, all of which led to his current faculty position. And in addition to being a fantastic scientist, he also received the Fred Wolf Mentorship Award for all the work that he's done mentoring students in informatics during his time here. So Vikas is going to describe his own work and then lay the stage for Dr. Predrag Radivojac, his talk, and Peja is a professor at Northeastern with a background in electrical engineering, in undergrad and master's, and then a PhD in computer and information science from Temple University, with his first two degrees being at Novi Sad and University of Belgrade. And his research interests align well with the general theme of the talk here, also in translational bioinformatics, combining computational biology and machine learning, looking at life at the molecular level and how this affects higher level phenotypes, and particularly looking at algorithms and analysis techniques related to the function of biological macromolecules, mass spec proteomics, gene interpretation, and precision health. So the full spectrum from molecule to patient. So with that, I will turn it over to you, Vikas.
Thank you very much, Pierre, for that introduction. And it's super exciting to be able to present some of the work that we've been doing recently. And as I mentioned early on the call, this is a nice homecoming for me, even though it's virtual, but it's a pleasure to present to this audience. So I'm going to talk about evidence-based calibration of computational tools for missense variant pathogenicity classification, but I'm going to spend a little time orienting everyone towards the broad field of variant pathogenicity prediction, function prediction, impact prediction, and then sort of give some historical context, talk about a few details about how these methods can be used in the clinical setting. And that should hopefully set the stage for Peja's talk, where they've undertaken a massive evaluation through community-wide experiments of these methods with different end goals in mind. I'll start with this classic paper from 1993 by Larry Hunter and Terry Klein. One of the earliest sort of attempts at this problem or this question was to be able to predict lethal variants in collagen genes, to be able to separate out variants here on the left that would be lethal versus non-lethal ones based off of sequence features. And if you're thinking machine learning AI, they used a decision tree and essentially wound up with this rule set, which I thought was very interesting, very fascinating, very inspiring paper actually. But the field has grown and has become more broad in the sense that there's a genetic question that's disease agnostic, where the goal is essentially given a sequence variant at the DNA level, incorporate some kind of information, be it evolution history, conservation, impact on protein structure and function, which I've shown here, and make a prediction of whether the variant is going to be pathogenic or benign. This could be a hard prediction of pathogenic or benign, or it could be a soft prediction where the predictor assigns a score between, let's say, 0 and 1 to classify the variant. This is going to be a very quick and poor attempt at kind of making a timeline of this field, but just to give you a sense of where the field was at, starting with that osteogenesis imperfecta paper. In the 2000s, there was a suite of methods that came out that were largely rule-based and unsupervised, extremely domain knowledge oriented, where empirical rules were derived based off of what was known about disease mutations that were collected until then, and then generalized to make classifications for unseen variants. Towards the end of the 2000s, a whole slew of supervised machine learning methods started coming out that use different features, right? So it could be conservation, as I mentioned, could be sequence context, protein structure, function, the whole gamut, essentially. And then more recently, there's been a family of methods that I would say that the field's kind of diversified, and there are several methods that have come out. But I think two broad developments. One was the emergence of meta-predictors. Meta-predictors are essentially methods that use outputs from these primary predictors as features. And then methods that went beyond just missense variant impact prediction. So I should mention that for most of this talk, I'm going to be focusing on missense variants. These are variants that lead to a replacement of one amino acid for another in the protein sequence. But there are methods that generalize to other kinds of variants as well. I just want to let everyone know about that. And as I mentioned, there's a large suite of tools. There's about 100 tools that addresses this goal. In this figure, I'm basically it's taken from VIPDB, where they've essentially made this word cloud of the different methods with citations being scaled. Sorry, with the font being scaled according to the number of citations the methods have accrued. So these methods find use in both basic research and clinical applications. And I should mention that they've been developed with very different design intentions in mind. Some of them try to predict the functional outcome, not necessarily disease or not disease. Some of them try to model directly whether the variant is implicated in disease or not. Some of them model specific aspects of function, like protein stability, for example. So they've all been kind of reused or reappropriated for the ultimate sort of question is whether a variant is capable of causing disease or not. We've contributed to this as well. A lot of my doctoral and sort of mid-postdoctoral work focused on developing a method called MutePred2. The key distinguishing feature of what we do is that we not only infer whether a variant is pathogenic or benign, we also make predictions about what biochemical or functional consequence could occur due to that variant. So inside MutePred2, there are 50 different machine learning methods, machine learning models that predict different structural and functional properties. And we make predictions based off of when the variant is actually when the protein sequence is actually unmutated versus when it's mutated, and then combine those two scores from each of these models to make predictions about which properties are likely to be disrupted. So that's sort of our contribution. And the method works great in practice. We've done evaluations, we've compared against other methods. But one's also able to derive functional signatures of different sort of disorders. So on the right here, I'm showing a data set of variants from de novo variants from neurodevelopmental disorders. And the take home of this particular image is that macromolecular mechanisms, macromolecular binding is a common mechanism that's disrupted in this disorder. And we also demonstrated that our predictions correlate with experimental values here. So in this case, for example, these are readouts from these two hybrid assays. This whole region in this picture is basically indicative of the fact that the protein STXBP1 loses interaction capability with its three partners. The mutate scores are high, and so are the predictions that that's the particular functional mechanism that's disrupted. Having said that, a lot of these methods have found great degrees of success across the board in the basic science context, but they've also been used in the clinical context. And here I'm again oversimplifying sort of the long history and the long story that sort of led up to this moment. But generally speaking, in around 2008, there were classification systems emerging, particularly starting in the breast cancer world, but then also in a more genetic sense from the ACMG and EMP. The American College of Medical Genetics and the Association for Molecular Pathology, for those who aren't aware. And these guidelines were the first step towards systematizing how different lines of evidence are used in the context of the clinic to be able to classify variants as being pathogenic, benign, and there are some other classes which I will get into in a little bit. In 2015, there was an updated set of guidelines, and computational methods featured more prominently. And I will talk about these guidelines in a bit. And this sort of sets up the rest of the talk, where a lot of this recent paper that we worked on, we were interested in asking how can we develop guidelines around the use of these tools in the clinical setting. To give a little bit of background about the ACMG-EMP guidelines, the guidelines essentially identify different lines of evidence or different data sources essentially for each variant. So a variant could have information about how common or rare it is in the population. One could have predictive model or computational evidence, as I just described before. There could be functional assays that are conducted in the lab that would say that if I introduce this variant in the lab, a particular function is knocked out, and other pieces of information as well. And the guidelines also organize these different lines of evidence in terms of strength. So there's very strong for pathogenic, very strong for benign, strong for pathogenic, strong for benign, moderate, supporting, intermediate. So there's basically nine strengths. Essentially, one could think of this as some notion of weights for each of these pieces of evidence. The guidelines also provide a set of rules. If I had two pieces of strong evidence for pathogenicity, what would I classify my variant as? So the variant would be classified into one of 5 different classes here: pathogenic, likely pathogenic, uncertain significance, likely benign, benign. In the context of pathogenic, we can just take one simple rule here, one rule set here. For example, if I had 2 pieces of supporting evidence and one very strong evidence in favor of pathogenicity, right, from these data sources, I'd be able to classify the variant as being pathogenic. So these are rules that are basically expert driven, and they've worked fairly well in practice. From the perspective of machine learning methods and computational methods more broadly, there's the PP3 and BP4 criterion. And the PP3 and BP4 criteria basically state that computational methods, predictive methods, serve as supporting lines of evidence, so the weakest strength of evidence. And there are other aspects of the guidelines that are kind of limited in the sense that there's not a clear sense of how these methods should be used. So for example, the criteria do not necessarily recommend which tools to be used. There's no recommendation on specific score thresholds. So if you remember, I did mention that many of these methods output soft classification, soft scores. And the assumption here is that the default output that the predictor provides is calibrated for clinical classification. And then there's basically an expectation that one would use multiple tools and make a consensus decision out of that. But you know, that it doesn't account for the fact that many of these predictors are correlated because they're often using the same training data sets, similar features, and probably have similar design intentions as well. So just to give you an example of that second assumption, right, the predictors are calibrated for clinical use, it's not necessarily the case, right? So if I'm going to pick two methods just to give you some sort of anecdotal reasoning. Polyphen2, for example, has a classification system where it assigns a variant as being either probably damaging or possibly damaging. And the way Polyphen2 developers decided on thresholds was to do a cross-validation, plot the ROC curves, plot the true positive rate, false positive rate, and then work their way backwards to which threshold gave them a 10% false positive rate and which threshold gave them a 20% false positive rate. Now, this data set, you know, this method was developed back in 2010. The data set was not necessarily clinically oriented in the way we would think of pathogenic benign variants today, or at least the way ACMG-AMP guidelines would think of them. Another example is from our own experience. So we did internal evaluations, and when we did internal evaluations, we defined our cutoffs as being at the 90% false positive rate and the 90% specificity and 95% specificity. So, you know, developers have a certain notion of how their method should be used. Does not necessarily mean that they calibrate for the clinical use case. In fact, there was a recent study where Benavenuta et al. Took a bunch of methods, ran them on ClinVar variants, found that the AUCs are really great, right? And it's over 9.9. But even though the AUCs are similar, each of these methods can be calibrated very differently. So if you had perfect calibration, you'd be in the diagonal line based on this picture. So calibration is sort of an important component, especially in the clinical context. So we collaborated over the last few years, we collaborated with the ClinGen sequence variant interpretation working group, particularly the computational subgroup, to basically answer three broad questions, right? So when can a prediction from some of these methods be considered clinically useful? What are the current practices in the use of computational tools, right? Can we go back and look at them? And then how do we come up with a quantitative framework that, you know, and that basically allows us to revise the current guidelines, but also be compatible with them without completely tearing them down, so to speak. Now, this is not easy, right? So this is different from, let's say, a typical, you know, assessment challenge or a calibration task, because many of these methods are binary classifiers. So they're trained on a set of disease variants and a set of non-disease variants, but they output soft scores that range between pathogenic and benign. And those scores have to be now mapped to 9 different levels of strength. And the challenge comes from sort of three, you can distill this challenge down to 3 questions, right? What does it actually mean to be supporting moderate, strong, and very strong? And, you know, can we quantify these? So the original guidelines use these terms, but there was no sense of, you know, framing this in the context of a number. Recent work from Taftigian et al. Basically had a very interesting solution, was to essentially use odds path or likelihood ratios as a quantifier of evidence strength. So under a Bayesian framework, you could use likelihood ratios as a means to say whether something is supporting moderate or strong. And I'm going to get into a little bit of the technical weeds here for a little bit, but you know, hopefully it'll become clear as we go through the slide. Essentially, when the original guidelines were framed, there was a sense that what being pathogenic really meant was that you had, if you were given a variant, you know, and you had some evidence around that variant, the probability that a variant was pathogenic given that evidence was at least 0.99, and for likely pathogenic it was at least 0.90. So that's sort of the framework where we can begin thinking about this in a quantitative sense. Likelihood ratios is a very useful measure in this context, because likelihood ratios connect the posterior odds to the prior odds. And the way Tuftekin et al. Basically developed their framework was essentially, model the likelihood ratio in terms of these different rules. So they said, let's assume the likelihood ratio is equal to some constant value, raised to these powers, and these powers basically are indicative of how many pieces of supporting, moderate, strong, and very strong evidence you have. And, at a particular, you can now set the prior, your left-hand side should be equal to one of these numbers. You can assume a certain prior, saying that on average, a variant is pathogenic 10% of the time, for example, and you can solve for the likelihood ratio here. I'm sorry, yeah, you can solve for C here because you can work your way backwards because you have both these quantities, you can get a likelihood ratio and you can solve for C. So the goal here is essentially find C such that, two pieces of strong evidence, for example, give you 0.99, right? So that's in sort of broad strokes. And they found that the value of C in this case was 350, and they basically proposed positive likelihood ratio values for each strength of for each criterion, right? So very strong would correspond to a 350, strong would be 18.7, and moderate would be 4.3, and supporting would be 2.08, and then you have the rest going on, so on and so forth. So the next question you can ask is, if you're doing something that's Bayesian, we need a prior probability for pathogenicity. In that paper, they assumed it to be 10% based off of some prior sense of how sequencing, clinical sequencing data emerged out of, basically pathogenic variants emerged out of clinical sequencing labs. We, in this paper, used a reference set. So we used population scale data. We used variants from Nomad and then estimated the prior in a positive unlabeled machine learning framework. So I'm not going to go into the details there, but essentially we found the prior to be 4.4%. So that revised our number. So that revised what very strong meant and strong. And because if you remember, it's dependent on the prior, right? So these likelihood ratios be dependent on the prior. And I also want to mention here is that for the rest of the talk, I'm going to go back and forth between likelihood ratios and posterior probabilities, because all you really need is that prior value at a given prior. These two are, you can compute, they're basically analogous, right? So I'm going to use them synonymously for the rest of the talk. Finally, how do we translate this framework into practice? And how do we, remember, our goal basically is to recommend a set of score intervals that would be clinically useful, and then based on that also make recommendations on which tools to use, because some of them may meet supporting evidence or may not. So we defined a new measure called a local likelihood ratio, local posterior probability, and develop an approach to calculate this. Again, I won't go into too much detail here, but at a fundamental level, we just changed the idea of saying, instead of saying the posterior is the probability of a variant being pathogenic given the evidence, like the evidence from the predictor, for example, we just frame it as what is the probability of variant is pathogenic given the score. So it's much more localized. And we denote this with a lowercase LR plus here. And the way we compute this is by estimating this on a known set of pathogenic and benign variants. So if you had a data set where you knew what the variants were classified as, you can run your predictor on that data set, get scores for each variant, organize it in this fashion, starting from 0. So let's just say hypothetically, we start from 0 going to 1, where 0 is benign and 1 is pathogenic. The variants would land in a certain fashion along these different scores. Then for each score, you take a localized window and then compute the fraction of pathogenic variants you got for that windows. And there's some adjustments that account for the prior here, but essentially you would keep doing this in overlapping windows and work your way from the highest score to the lowest. And that way you can generate now for each score a local posterior probability value on the y-axis, and on the x-axis is the score. And we already, from the previous table I showed you, we know what the line for strong, very strong, moderate, and supporting should be. So whenever this curve intersects that particular line, we can claim that that's the score at which we would get very strong or strong, moderate, or supporting evidence. You can do the same for benign variants, and thus you'd be able to partition the space of scores into these 9 different intervals, and we, one would be able to assess how useful the tool is in the clinical context. So we did this empirically. I won't go into the details of the data collection here, but we extracted data from ClinVar, went through a bunch of filtering steps. Most notably, we made sure that none of the variants that we were evaluating on were in the training sets of many of the tools that we were assessing. We assessed 13 tools. Not all of them are supervised machine learning methods. So there's no notion of trainings. In some cases, we didn't have to do any filtering. The breakdown was basically like that. We had about 5,000 likely pathogenic variants, and a little over 5,000, maybe close to 6,000 likely benign variants. So we ran through our algorithm, and essentially we generated a local posterior probability curve. So I'm showing you one example here for a tool called Revel. The x-axis here is the Revel scores goes from 0 to 1. The y-axis is the local posterior, as I mentioned. Each of the horizontal lines corresponds to the strength. There's two curves. The black curve here is the point estimate, and the gray curve here is the one-sided 95% confidence bound. So it's a little more stringent. It's to the right in the case of the pathogenic, and to the left in the case of benigns. So as you can see in Revel here, it crosses the supporting at around 0.6, at around 0.75, 0.76-ish, you can get moderate evidence for pathogenicity, and close to 0.9 can get strong. So the computational methods can actually provide strong evidence for pathogenicity, and the same holds for benign as well. Another example, we can look at another example, and in this case, I'm showing you CAD, where the score basically, as the score range actually goes from 0 to well, in theory, I guess infinity, but in this context, at least in our data, it goes as high as 64. The curve basically passes the supporting and moderate lines, but then flattens out after that. So you could at most get moderate evidence for CAD if you applied a threshold of 25.6, 26. But on the benign side, CAD easily reaches strong levels of evidence as well for benignity. So we did this for 13 tools, and I'll show you the results in a bit. One thing we did want to explore was whether the default thresholds of some standard methods that are very commonly used in this context, if they would, achieve supporting levels of evidence. And it turns out they don't, right? So these are the default thresholds that one would use when one would use SIFT, Polyfen, or CAD. And the local likelihood ratios don't, or the posterior probabilities don't match the ones I showed in the table earlier. So the evidence strength basically was not met. So among the 13 tools that we ran, we basically have identified score thresholds for each of these tools. We've reported these. It's all in the bioRxiv paper. But the key point I want to make is there are 4 tools that give up to strong evidence for pathogenicity, and 3 that give strong evidence for benign. And the next exercise we undertook was to sort of validate these. I mean, but this is more of a, just of a sanity check. What we did is we let variants accumulate in ClinVar and then extracted a subset. So basically we call this the ClinVar 2020 set. So this is the data set that was not used for our calibration process. So it was variants that accumulate in ClinVar after we did the calibration. And the filtering steps were all very similar. We filtered against the training sets. We filtered against our calibration set as well. And we basically asked if we were to use the thresholds on face value, would we be able to get the same likelihood ratios back, or at least or higher on the ClinVar 2020 set. And this is a busy figure, and I'm just going to focus on maybe 2 to just illustrate sort of an example. So in the case of Bayes-Dell, the likelihood ratios for strong, moderate, supporting were 52, 7, and 3. This is much higher than the ones that I reported in the table earlier. But on a data set of nomad variants, basically population variants, only 1% of those variants, or 1.5% of those variants, were predicted to be pathogenic at the with strong level of evidence. This is much lower than the prior that we estimated, so it seems reasonable that way. And the one point I want to make with these 2 pink boxes is that, of course, the choice of tool involves a trade-off, right, between evidential strength, right? You could pick a tool that will give you maybe a high basically a high likelihood ratio gives you strong evidence, but it would mean that not many variants would get classified in that interval, right? So if if one were to pick something that was providing, let's say, moderate evidence, you would end up with 10% variance from the NOMAD set. And NOMAD is basically genome sequencing data from seemingly healthy population. So there is this trade-off involved. We presented based on our findings, we presented some updates to the 2015 ACMG AMP guidelines, specifically the PP3, PP4 criteria. We claim that methods can be used beyond supporting. The computational predictors can provide moderate and strong evidence for both pathogenicity and benignity. We also argue for a choice of a tool to be made before its scores are seen, in the context of a variant classification. So you pick one tool, sort of stick with it. And ideally, it should be something that has at least strong evidence for pathogenicity and moderate for benignity. And we thought we didn't test the use of multiple tools. That's just one of the key points of the PP3 PP4 criteria. We didn't test it systematically, but based on whatever we've seen from our preliminary analysis, we recommend using a single tool rather than an uncalibrated consensus, because it's really hard to say whether these tools are correlated to begin with and what the effect of these and how to calibrate all of them in parallel. It's a challenging question. So I'm going to summarize and maybe open the floor for questions. The key take home is that developer default recommended thresholds are not always calibrated for clinical use. And I think this holds broader than even the context of genetic variant interpretation. And in this case, computational predictors can provide more than supporting evidence. And we recommend using a single tool without any access to the results, right? So before you see prediction results and before you ideally see other lines of evidence, you want to make the choice of that one particular tool. We do realize that now that computational methods will have a more important role, our findings are basically indicating that these can provide stronger levels of evidence, so they'll have a bigger say in whether a variant becomes pathogenic or benign. That changes the game a little bit, in terms of training new models and even assessing new methods. So we recommend that rigorous documentation be associated with each variant, right? What tools were used, what score thresholds were used, so on and so forth. From a methodology standpoint, local posterior probability is an interesting new approach to assess and calibrate predictions for clinical use. So in this context, we used it for computational predictors, but in theory, it could work on any continuous distribution, right, of scores. So it could be scores that come from a functional assay. We could map that to the ACMG AMP guidelines using this approach. And we could extend this to gene-specific, disease-specific context. Here, we kind of took variants in a disease-agnostic fashion as a bunch from ClinVar because we wanted large amounts of data. Some caveats, ClinVar annotations often incorporate prediction methods, and we don't have access to that level of detail, to my earlier point in the discussion slide. There's definitely overlap between information used by predictors and other lines of evidence, and that's something that is an issue, if you're thinking about the ACMG AMP guidelines in general. There's some technical aspects, for example, whether when we estimated our priors, is the data set representative, what are the, there are limitations to the ACMG rules and the Bayesian framework itself. And a lot of what we've done is developed in the context of the ACMG AMP guidelines, may not behave as expected in other contexts, but it certainly is a way to think about this sort of beyond genetic variant interpretation as to how one could think about clinical computational methods being used in the clinical context. So with that, I'd like to thank our collaborators, the SVI computational subgroup members, and some of the predictor developers who actually gave us predictions and even helped us filter out data from their training sets. There's a preprint on bioRxiv. The paper has been accepted, and we're just doing our final rounds of revisions, and we should be out there soon. My group is looking for postdocs in variant and genome interpretation, EHR-based phenotyping, applied machine learning. So for more information, please feel free to reach out to me. And if there's someone who's looking for a position, please let me know.
Thank you, Vikas. That was outstanding, and finished on time and actually a little ahead of schedule. So that's great. So before what we planned was allowing a little time for questions for Vikas. Then we'll have Peja present some questions for Peja, and then questions for the group. So, any questions for Vikas? You can type it into the chat, or you can unmute yourself and ask the question verbally.
Vikas, this is Joe Colarafi. Thanks for your presentation. Can you hear me?
Yes, I can. Hi.
I teach biostats here at the School of Medicine in Spokane, one of the UW medical schools. Did you use any clinical or comorbidity variables for your models?
No, not we so we did not use anything in this context because they are considered as separate lines of evidence in the context of genome. I mean, am I understanding the question right? Or
I think so. Yeah.
Okay. Yeah, in this case we didn't, because we focus solely on the context of thinking about computational methods and improving those specific PP3, PP4 criteria in the ACMGMP guideline.
Is there some reason that you wouldn't consider doing that with future research? I mean, is there some, it seems like it would add to the sensitivity, at least.
It would. So I guess there's a broader question, which, I mean, it's worth sort of discussing is, you know, the ACMGMP guidelines have this way of framing, basically putting each source of information into a box, right? Saying this is population evidence, you know, co-segregation, clinical variables, so on and so forth.
Mm-hmm.
There may be value in thinking about modeling the whole set of guidelines with new information, right? So sort of kind of starting from scratch, where one were to integrate evidence from different lines and add clinical covariates and comorbidities to that. But right now we're strictly preserving ourselves sort of to, you know, again, one of the points I made earlier was to kind of not just do something that was a machine learning evaluation or assessment, but also kind of obey or fall in line with how the guidelines were framed in 2015. I don't know if in the future, if the guidelines, I know that they're looking to make changes and there'll be an update sometime. I don't know if one of the things that they're thinking of doing is precisely what you're mentioning.
Okay.
I'd love to read those conversations.
Because I don't know, but what I would say is you know, this is a recurring challenge within biomedical data science, which is what you're using as a gold standard is a non-evidence-based, non-statistically based consensus statement by a bunch of geneticists sitting around a table figuring out how they determine pathogenicity or non-pathogenicity, which is far better than an individual doing it without the guidelines or individuals doing it each with their own guidelines. So I don't mean to diminish it, but where it creates a challenge is that it is possible your machine learning algorithms, particularly, Joe, if you start including some of the clinical stuff, will outperform the ability of the human annotators to do this. And then how do you assess the performance of something that's performing better than your heretofore gold standard of a consensus opinion amongst multiple human annotators? So I think it's a very fascinating question. And I think what you've done is very appropriate because it allows people to look at the data in multiple ways, including against what we consider the gold standard today. So there is a question in the chat box that says, given a presenting condition that's very pathological from a clinical perspective, but evidence from, I mean, it says test tube studies, but presumably bench work, lab animals, computer models, suggest the condition shouldn't present as pathologic as it does. How do you reverse engineer the mismatch? Or how do you determine what the underlying issue was with your predictive model?
That's an interesting question.
Or I'll reframe it. Models predict it's not pathogenic, presents as pathogenic. How do you address that discrepancy?
So I think part of it also has to do with what kind of clinical actionability comes with this information, right? So I mean, I'm not a clinical geneticist, so I wouldn't know exactly, but from what I understand, more often than not, making a pathogenic classification is more of a diagnostic process. And I don't quite know, at least as far as my reading is, I'm not very familiar with what outcomes, you know, how well outcomes associate with some of these classifications. I think for some disorders like breast cancer, there's like good literature out there. And I think it begins with that, right? So how does one reverse engineer, you know, as Peter mentioned, it presents as pathogenic, but it's not called pathogenic by the predictor. There's actually many possible reasons for that. One is that at some level, all these methods are trained on function, right? So they are basically modeling, you know, the impact of variant on protein structure and protein function. It may very well be that the impact is, you know, it's a heterozygous state for that variant, right? So there's a perfectly functional protein that's floating around in the cell, and that's just enough to basically preserve the healthy state. There might be other factors as well. For example, the gene may not have anything to do with the disorder, or more specifically, like if the gene has multiple functions, maybe that particular function that we are very good at modeling, right? The disruption of function that we're very good at modeling has nothing to do with the disease phenotype. And I think you'll see a little bit of this in Peja's talk, that functional experiments also don't necessarily line up that, you know, neatly with phenotype. But yeah, it's a good question. Reverse engineering, you know, going, you know, how, I think it's one of those things is the way the guidelines are set up. It's not purely a computational task. I think we'd have to kind of go back and see if, you know, if there was, again, is the variant, you know, if penetrance was an issue, right? So there's a lot of information that one would have to go back and see. I think the key thing, at least one of the things I wanted to mention in the talk, was it'd be nice to record all of that information as to like why something was asserted as being pathogenic for the time being. I don't think we're quite there yet.
Great. Well, thank you, Vikas. Peja, why don't you go ahead and give your presentation, and then we'll do about 5 minutes of questions for specific to Peja's talk, and then we can turn it open until 2:30 for wide open questions. Thanks very much.
Thank you, Peter. My name is Predrag Radivojac here at Northeastern University, and my goal for today is to expand upon what Vikas discussed, basically to incorporate other types of problems in the field of genome interpretation, to discuss the perspectives of other stakeholders, and also to talk about the role of KG, which is a community experiment in this field of genome interpretation. And I'll give it an attempt to define what that field might be, just for the sake of us having this exercise. And you know, one way to think about it is that this is a development and application of analysis and prediction techniques that are aimed at elucidating the impact of variants in an individual genome on different molecular, cellular, or organismal phenotypes. And as you can see, just based on that sentence, it's a mouthful, and what goes right from it is this remarkable breadth and complexity of this field. First of all, we have different types of phenotypes, from molecular effects to all the way to high-level phenotypes. You know, we have different biological and environmental contexts. There's a lot of different data modalities in this field, and also different data generating platforms. Even for the same type of data, you might have a low throughput and a high throughput experiment, and all of that plays different roles. And then there's a lot of different computational techniques in this field that we need to sift through and figure out how they work together. And of course, my goal will be to talk about the evaluation of computational techniques. That's why it's a bolded word here, and their application. And of course, this field has a lot of ethical challenges, both in the research process, but also in data sharing and deployment of these methods, such that these methods, when applied in a clinical setting, would be accepted by the society. And so we run critical assessment of genome interpretation, which is, we call KG. And this is a challenge founded around 2010 by John Molt and Stephen Brenner. Basically, it's a series of different rounds of challenges modeled just like CASP, where the organizers go through a process of challenge development. Then once the challenge is posted, for example, it could be to predict the impact of missense variants, but it could also be to predict whether a person whose exomes are provided have a particular disease. So it could be a very large range of these types of things. And then we go through the prediction season where different predictors are submitting their predictions. And then after that, there is an assessment season where the assessors actually do not know. So it's a blinded prediction, so they don't know who the predictors are. They assess the performance of these predictors. And finally, there is a dissemination of these results through conferences, publications, and what we hope to be KG calibrated reference. So the mission of KG is really to conduct these community experiments, assess state-of-the-art in computational interpretation of genetic variants. And we have also various objectives in this whole thing. Obviously, to evaluate the accuracy is our very important challenge, not just to evaluate the accuracy, but also to select the accuracy measures that will be used in different communities and that will make sense for different types of tasks. Of course, to give credit to predictors and highlight what did they do well, reveal bottlenecks in the field. As Vikas just presented here, we want also strive to contribute to the development of guidelines in clinical practice. And I will show you a challenge that was a precursor to the study that Vikas has conducted. Of course, we are a forum for dissemination of research results. And of course, we promote ethical practices and responsible data governance. We have three primary communities in mind, but this is not a closed set of communities. Clearly, we are there for method developers to give them feedback and to discuss what works and what doesn't work and how to measure accuracy, what is the novelties in the computational world and so on. Clinicians are another set of stakeholders, as Vikas just discussed, so I'm just going to skip that part. And finally, we have also biomedical researchers who might be designing maybe new enzymes with particular new functions, and they might actually have different objectives in mind. And as AI techniques are gaining more and more attention, it is probably wise to think that regulators are going to be another audience of this type of experiments. And as I mentioned, we have many different settings, and I'll talk about that a little bit more. Rare disease, complex traits, cancer. Sometimes we predict the response to drugs for variants. We deal with single variants like amino acid variants or single nucleotide variants. We work with gene panels, exomes, and whole genome sequences. Again, high throughput and low throughput experiments with their strengths and weaknesses. KG challenges have addressed different population groups. We had a cohort from Germany and Italy with different patients. We also had a cohort of African Americans. And so we try to provide benefits for all groups. Sometimes we also have auxiliary data, which is a real life challenge here, that sometimes we have clinical measurements, omics data, and so on. And in certain cases, we also dealt with non-human data, if only for the purpose of learning that non-human data is not of such a large interest as human data. So participation has been limited in these types of challenges. A little bit about participation in KG. As I said, it's a community challenge. And from KG1 to KG5, we had about 450 different participants, and they come from different countries. And this is just a brief overview of these individuals from the world. And then I can show you also in the United States how this participation looks like. The state of Washington doing well, California, Massachusetts, Maryland, Texas, basically as one would expect in these types of research. So let me talk a little bit more about now KG and how we do things. and go into the weeds of genome interpretation and trying to unify different ways to assess the accuracies of interpretation. So, as I said, we go through different phases: a challenge development, prediction season, assessment season, and dissemination. As you can see here, the first 5 kgs, we had the number of groups and the number of submissions. These numbers are very reasonable. Just to say that KG6 that was just completed, these numbers are roughly doubled from KG5. And so that bar would sort of chain the other bars almost together. And the scale and diversity of these challenges is also shown in this pie chart. So for example, on genomic scale, we can deal with genomes, exomes, and gene panels. And phenotypes can be complex trait, cancer, rare disease, etc. I will also talk about a few challenges that are specific. One is a NAGLU challenge, another one from KG4. I will also talk about the P10 challenge from KG5. I'll talk about Crohn's disease challenge, which is a complex phenotype disease from KG4. And I'll also talk about the annotate all missense challenge, which is a challenge that is directly related to what Vikas presented here. So let me get in that detail. So annotate all missense challenge was run in KG5. This is one of the challenges that was the least related to CASP. Rather, it was related to other challenges where predictors deposit their predictions to KG portal and also in a database called DBNSFP. And at that point, we wait for the number of variants to accumulate. And that wait period was more than a year. In this particular case, it was about a year and a half or so. And then once we have the gold standard, the variants, in this case, both from ClinVar and HGMD, we have obviously removed variants that existed at the time when predictors deposited their predictions, and then we could evaluate it. And on the left-hand side, you can see that this is a typical machine learning evaluation that we call on high confidence variants, which are variants that are pathogenic in ClinVar and DMs in HGMD. And on the right-hand side, we have pathogenic and likely pathogenic in ClinVar and DM question mark in HGMD. But I'll focus here on the left-hand side. And as you can see, this is a very good data set and the curves are smooth. And the idea was, okay, let's just do a regular presentation of results as an area under the curve. On the x-axis is a false positive rate, on the y-axis is the true positive rate. And here we present the top 2 meta predictors, which were Revel and Meta LR. And the top 2 for this particular type of challenge, sort of independent or single predictors or primary predictors, VEST3 and eigen. As you can see, they have very similar performance, with Revel being a little bit better than others. And as a form of comparison, we have PolyFan2, which is a method that was developed in 2010. I have to apologize. Just a second. Give me 1 second. Okay, so let me try to focus and explain briefly that in this particular case, we see that the performance is good and the machine learning community would be happy because these numbers are roughly in the 90s. However, this is not the most important part of these prediction challenges, because clinicians would, okay, I'm really sorry, let me address this. Because the clinicians would be interested in low levels of false positive rate and as high as possible levels of true positive rate. And therefore, this part in the left-hand part of the ROC curve space would be really of importance to them. So I'm going to plot here the truncated ROC curve, which actually gives us the false positive rate up to 0.2, so only 20%, and true positive rate is plotted in the full interval. And now we can see a little bit better how these curves look like up to 20% false positive rate. So this is a more relevant region for the clinicians because obviously we are allowing a smaller number of errors. And so if I plot what exactly here is the random predictors, you can see this dotted, gray dotted curve or line. These predictors are way better than random, and obviously these predictors are doing well. And as Vikas just explained, they're actually providing a very good evidence in the clinic, and they are underappreciated. And so here we have a different way to assess that. We have different types of databases that play the role, and we have a much larger set of variants. Now, how does all of that look when we talk about the clinical setting? In a clinical setting, as Vikas just described, I'm going to pick here one predictor only, which is Revel score. And I'm going to show you this particular probability of pathogenicity, which is the posterior that Vikas talked about. And so here, I'm showing you what is the influence of different priors on that posterior. Vikas has estimated the prior to be 4.5%, but a prior of 10%, which would be represented here by this particular posterior curve, this prior of 10% was actually determined by clinicians just observationally in the clinic, that about 10% of cases that are seeking etiological diagnosis, that they would be finding a variant in 1 of 10 cases. And so in this case, we can see that this posterior can go all the way over 0.9 when prior is 10%. And that is really bigger and larger number than 90% posterior that ClinGen, or rather ACMG and AMP guidelines prescribe for the variant to be called likely pathogenic. Therefore, predictors here under these types of conditions already should be able to outright provide the evidence that this variant is pathogenic, but of course ACMG and AMP criteria do not allow for that. However, keep in mind this, the predictors actually have very good performance. This is, of course, missense variance. However, if we take a setting that might be called a screening setting, let's say that we are not aware of any pathogenic variants in this person's genome, in this case, the prior might be 1%, and in this case, these predictors are actually achieving lower performance. And so we have to be very, very careful for which task we are applying this predictor and how we are going about this prior. And so just a note that this number here, this particular threshold here means that at a threshold of about 0.8, this model Revel would predict for about 4% of variance to be in NOMAD, a rare variance in NOMAD, to be having strong evidence. About 10% would have at least moderate evidence, and 14% would have supporting evidence. And so these thresholds are here in red, blue, and green. Now, if I map that back to the ROC curves, you can see that for Revel and a prior probability of 10%, where these particular lines of evidence are. And so indeed, they're all on the left-hand side, and this is for the prior of 0.1. So roughly false positive rate is below 8%, and here it's for supporting. For moderate, it's roughly about 4%, and for strong, it's roughly about 1%. Now, if I take into consideration slightly different challenge, so let's not worry just about pathogenicity and expand it to predicting some other biochemical effect. And here I'm talking about the NAGLU challenge, in which we had a set of 163 variants for NAGLU gene involved in a rare recessive disorder and trying to predict fractional enzymatic activity of these variants. And on the right-hand side will be a P10 protein where we are predicting a biochemical effect related to protein stability. So this is a measurement actually out of University of Washington from Doug Fowler, Elias Tarita, and the VAMP-seq method. On the left-hand side, you can see that on the x-axis, we have the prediction of performance of this of fractional enzymatic activity. And on the y-axis, we have the observed enzymatic activity. And so this agreement is moderate. So there's a lot of scatter in this plot. And these methods, even though they can provide clinical evidence on a discrete scale in terms of supporting, moderate, and strong, are actually not as good in predicting actual biochemical phenotypes. And so there's an area of calibrating these scores to provide biologically or maybe biochemically meaningful outputs that these methods still have to provide. And in terms of this particular method, in this case, this is actually Vikas's method, MutePred2, that was the best ranked method in this challenge in KG4. We know that the prior for this particular gene, NAGLU, is 25%. Why? Because this experiment considered a random sample of variants from his exact database, and therefore the number of variants that have performance or enzymatic activity below 15%, which our collaborators determined strongly correlates with disease outcome. And so the number of those variants is about 25%, and you can take it as a class prior. So a point in this challenge is that the level of biochemical activity in this case correlates with the clinical outcome And so the same predictor can in principle be used to give a prediction between for exact relative fractional exome activity, and then also to be considered for the clinical performance. And if you look at this lower left-hand corner, these are the predictions where the method predicts roughly 0% enzymatic activity, and the true values are also around 0. So in this one corner, the prediction method is really, really good. Similar can be said for Fox-Chase algorithm here on the P10 challenge, but as you can see, there's a little bit more scatter here, and this method in this case achieves only supporting and moderate evidence. And again, there's a particular level of correlation between the outcomes between protein abundance and predicted activity. These are the ROC curves. Already I have explained those, and so the performance looks good again when you discretize the prediction, but when you look at the relative fractional activity or relative protein abundance, it's not as good. Now, of course, whenever we predict continuous phenotypes such as enzymatic activity, we can use other types of measures, and KG has never committed to a single measure of performance. It rather gives an array of different measures that are relevant in different situations. And so one of them is ROC performance, which is useful in machine learning settings. One is here, where we give different types of correlation, Kendall's and Pearson's correlation. And another would be the clinical types of performance that Vikas just talked about. So you can see here that Pearson's correlation is roughly about 0.6. And somehow still a lot is to be desired in this type of experiment. So there's still a lot of room for improvement. And just let me say that this 0.97 is actually experimental reproducibility of that assay. These are the technical replicates. And therefore, it's not quite clear whether how much room there is, but we believe there's still plenty of room for improvement of these methods. And again, here one can use the setting and framework that Vikas just described, except that here the axes are reversed. So the low prediction means low enzymatic activity, and the blue curve here would provide the likelihood ratio, the LR plus, local LR plus that he explained. And the red curve here would be the probability of pathogenicity or the posterior. So these can be done on a single gene if we have enough data. Here for NAGLU and for P10. And if we don't have enough data, the average predictions obtained on a large set of genes, just as Vikas has presented, would be a way to go. Now, there's a lot of diversities in these methods, and KG has worked quite a bit to understand their performance. There are biophysical approaches, there are structure-based machine learning approaches, sequence-based approaches. And one thing that emerged through a lot of missense challenges in KG experiments was that structure-based approaches, by and large, although sometimes they performed well, they have not really outperformed sequence-based methods. We do not really know why this is, but I also should say that this is the situation before AlphaFold, when finding real good structural information was difficult, and a lot of variants could not be mapped to protein structures. So we will see now with the new landscape and AlphaFold predictions, whether in the next iteration of KG we will have different results. Another interesting thing with these methods is that sometimes these methods have surprisingly correlated performance. Even methods that come from maybe a sequence-based predictor trained in a supervised setting and an unsupervised model, such as the one from Olivier Lichtarge's group called evolutionary action. Again, these methods are trained or developed to predict the same outcomes, and even they have done that in such different ways, these predictors are quite correlated, and this is yet another argument why a simple combination and a consensus of the predictors should not be sought, especially not for clinical performances. Now, let me briefly discuss other types of challenges in KG. We had prediction of course, coding variants that I just described, but also non-coding. And so we had splicing and regulatory variants. And this is just one experiment of called regulation saturation from KG5, when a different set of promoters, 9 promoters, and I believe 4 different enhancers were used to understand the impact on the expression levels. And of course, the purple circles here annotate the ones that have, in this particular case, decreased expression, and here increased expression. As you can see, the performances in the ROC space, when we quantize those values, are decent, not great. In terms of correlation, these correlations are quite decent as well. Now, notice that even though I'm saying here, okay, the ROC is only about 0.81, if you looked at the LR plus, that value would be around 4 to 6, actually, in the high range of the prediction scores. Therefore, we believe that even in the regulatory effect challenges, such as splicing and regulation, these predictors actually have signal and could be used for more than what they are used today. And now, if I go to enhancers, enhancers work a little bit worse than promoters, and I will go fast through this challenge and just say that predictions are, in both cases, these are the top two methods, ensemble and surf, about only 60 and 64% for prediction of increased activity, and a better performance, as also for promoters, is the decrease in activity. So this is where predictors work better. Finally, Pearson's correlation around 0.5. Now, moving on to yet another type of challenge in this huge field of genome interpretation is prediction of complex phenotypes. And so this was an early challenge from KG4 on Crohn's disease, where we had around 100 and so patients, where 64 were cases and 47 were controls. Those were exome data, all of German ancestry. And the goal was, given these exomes to the predictors, can they predict which of these individuals had Crohn's disease? So this was one of the 3 Crohn's challenges in succession, where the first 2 always had some kind of problems, either with population structure or batch effects. And this was a challenge in which we are unaware of any kinds of problems of that sort. It doesn't mean that they don't exist, but we have not found them yet. And so, as you can see, performance in this range is around 72% between top 2 methods, which were Maryland and London. And when you describe the distributions of scores between cases, which is the red curve here, and the controls, one can see a reasonable separation between the scores. And importantly, mapping this to the same framework of local LR plus that Vikas just explained, you can see that again, we see values of around 6, which means that these particular predictions could be useful in the clinic at almost the same level as some of the computational predictions for missense variants. We think really this is one of the important parts of our studies in KG, that we are putting all of these different types of problems, from missense variants to complex phenotypes, into the same framework and giving roughly the same types of measures for all of these challenges, and therefore concluding that there's value in these types of predictions. And of course, this Crohn's disease challenge was a precursor to polygenic risk scores types of prediction. And in KG6, we have actually ran one of such challenges on more than 3,000 patients, if not even 5,000. And the results of that challenge will be available soon. So briefly to conclude, I am running out of time. In KG6, we ran it in 2021 and 2022. This is almost concluding. We had 30 new challenges, hundreds of participants, 71 groups from 17 countries. And we are currently in the middle of producing a special issue for this challenge with some very interesting results. One of them is the prediction of rare diseases and diagnostics, where predictors were able to improve on the diagnosis for 2 out of 15 different patients who were previously undiagnosed by the state-of-the-art pipelines at the Rare Genomes Project here at Broad Institute. And these predictors also on some data where we actually did not. We did have diagnosis, but withdrew this from the predictors. They actually performed pretty well. And so what are the other ongoing activities in KG? So just to start concluding is we are undergoing various educational activities. Just past month, we have had a MLD bootcamp, which is, I believe, monochromatic leukodystrophy disease, where the idea was to predict the the fractional enzyme activity of the ARSA gene. But we ran this as a bootcamp for undergraduate students, taught them genetics, machine learning, and programming. And after 2 weeks, the goal was for them to submit the predictions to this ARSA challenge. And ARSA challenge is not closed, so we have not fully evaluated that yet. So if there are any interested individuals on this call, ARSA challenge is open until November 15th. And it's a very interesting challenge. We have about 280 variants for which we have resolved fractional enzyme activity. Another KG activity is that we will run an invited session at ASHG. So join us in Los Angeles on October 25th at 4:30 p.m. local time. And also we are recruiting a new KG scientist. So what are the contributions of KG? We believe that KG as a challenge that includes the world and levels the playing field with different methods and addresses multiple audiences provides basically a picture of the current state of the art in interpreting the impact of genetic variants. Some of our challenges provide a basis for the development of new methods and also for calibration in clinical settings. We also are developing this active community that is both competitive and friendly in jointly addressing these problems. And our principal findings relate to what Vikas has just described. We believe that computational methods are already more reliable than currently recognized. Obviously, missense variants in particular, but not just missense. It also holds through cancer. It holds for the variants in regulation and splicing as well. We find that, and even in complex disease, we also find that evaluation is a very tricky part, with different communities having different concerns and different needs with respect to evaluation of these challenges. And the framework that Vikas has described and that I have been sort of further emphasizing here presents a unification throughout all of these disparate types of challenges. And we believe that this is important. This will allow these different communities to actually talk through these types of evaluation and be able to understand the contributions that these different fields are actually providing. Finally, I will say that there's still a lot of obstacles in data sharing and ethical challenges that are preventing us from fully exploiting all the data available and engaging all individuals who are interested in this problem. And as KG, we are developing data use agreements and trying to streamline this process such that clinical data can be responsibly shared and that other types of challenges are also overcome. And one of the ethical challenges we always warn about is overinterpretation of results, especially when this type of overinterpretation can lead to problems in the social context. And so with this, I will just thank many contributors of KG. KG was founded by John Molt and Stephen Brenner. John Molt has left this effort and I have succeeded him about 4 years ago. So I'm one of the other co-chair of KG. Multiple KG scientists, Susanna, Roger, Gaia, Daniel, and Tina Bakolica, who is listed here on the right-hand side. You know, for many of these things in this paper, all the analyses were pretty much performed by Shantanu Jain, a postdoc in my group. And the preprint with summary of 10 years of KG challenges and a lot of results is actually provided on arXiv, and this paper is in review, and so hopefully it will be accepted soon. And so with this, thank you for your attention. I was very excited to talk here at the University of Washington, and again, welcome any questions here.